Good morning, my dear students. In my today's class, I am going to discuss about the process of rDNA technology. Now, this topic I am dividing into two. Perhaps in the half of the lecture, I will be concentrating mostly on the process involved in the preparation of recombinant DNA, and of course, in the rest, the second half, we will be concentrating on how biotechnology could be used for the different beneficial aspects of humankind. So that aspect also I will be giving some importance. Okay, so this is what I am planning to do in this lecture. First one is a process of recombinant DNA technology. Now, this involves the following steps. It involves several steps in a specific sequence. So, one cannot be skipped and one cannot be overlooked also. So, everything I have just put in a specific sequence. First one is isolation of genetic material in the form of DNA. First one is isolation. The second one is cutting of DNA at a specific location. This is the second one. The third step is the amplification of a gene of interest using the PCR, polymerase chain reaction. Then the insertion of the rDNA into the host cell or the host organism. The insertion. Amplification is the third step. Insertion is the first step. Obtaining the foreign gene product. Obtaining the product is the fifth step. Then downstreaming process. That is the last one. Okay, so these are the 6 steps involved in the DNA technology. Once again, before proceeding one after another, let me give once again the steps to you. The first one is isolation. Isolation of what? Isolation of the genetic material. Second one is a cutting of the genetic material. Third one is amplification of that cut portion of the DNA. Then insertion of this rDNA in the host organism. Then obtaining the product. Then finally downstreaming the process. So these are the different steps involved. Now I am going to take up one after another, as I have already told you. First one is the step on isolation. Isolation of the genetic material. Okay, now how will you isolate a genetic material? The DNA, the genetic material in majority of the organism needs to be in pure form. If the DNA is to be cut with the restriction enzymes, so the first condition number 1 is it should be in its pure form. If it has to be cut with the restriction enzyme, otherwise restriction enzyme will not be able to cut the genetic material. So if a cut has to be made, then it has to be in the pure form. So this is a condition number 1. Okay. DNA along with other macromolecules such as RNA, proteins, polysaccharides and lipids are released by breaking the cell open. This can be achieved by treating. So, see, in a plant cell, along with the DNA, what are all the materials you are getting? RNA will be there, proteins will be there, polysaccharides will be there, and then lipids will also be there. So, these are all the other substances. You can call it as impurities also if you want. If you are going to call as the DNA is a pure form, these are, when compared to DNA, these are only impurities. For a DNA, these are all only the impurities. So these 4 structures which are found mixed along with the DNA has to be got rid of. How to do that? So the cells have to be treated with in the suitable way. This can be achieved by, if you are going to carry out your work in a bacteria, then with lysozyme. The plant cells are treated with cellulose, then fungus with chitinase. See, if they are released by breaking the cell open, and breaking this is achieved by this process. Okay, so this restriction enzyme, it is how this is done. This restriction, this is the R plasmid. Restriction enzyme opens this out, and then the DNA ligase is hybridizing it. This is the original plasmid, this is the ligated one, and then you get it in the, this is a foreign DNA, and then you are getting it there. The overall summary, that slide is only a overall summary. Okay. Now the genes are located in long molecules of a DNA intertwined with the proteins. So a gene will be always seen along with the protein. RNA can be removed by the treatment with ribonuclease. Proteins can be removed by proteases. Other molecules can be removed by appropriate treatments. You see. So what are the different materials that you get mixed along with the DNA? You will be getting RNA, you will be getting proteins, and you will be getting other molecules also. RNAs can be removed by ribonucleases. Proteins can be proteases. Then other molecules can be removed by the suitable techniques, appropriate techniques. So by using the different techniques meant for this, you will be able to get the pure DNA. This purified DNA ultimately precipitates out after addition of a chilled ethanol, which can be seen as a collection of fine threads in suspension. So in a test tube, perhaps you will be able to see them as a fine threads. Finally, when you remove all the unwanted structure things. What is the unwanted structures? The RNA, proteins, and the other molecules. If you are able to remove, then when you are adding the chilled ethanol, you will be able to see them as a fine threads. So that is a step number 1. That is what is known as the isolation. Step number 1 is isolation, I told you. The second one is cutting of DNA at specific locations. Now restriction enzymes or enzymes which are called the endonucleases, they are incubated. Their digestions are performed by incubating the purified DNA molecules with or nuclease at optimal conditions for that enzyme. Okay, so each enzyme will be having a optimal, a very narrow range in which they will be very active. You must be knowing about that. Any enzyme will be very specific, and it has got a very narrow range for its optimum expression or for optimum reaction to, or the optimum output to give. Okay, so this is done by incubating the purified DNA along with the endonucleases, and then it will be able to, what is known as at the optimal temperature, if you are incubating it, you will be able to get the product. Now this product, this agarose gel electrophoresis method is used or employed to check the progression of a restriction endonuclease digestion. So this digestion process is monitored by what is known as the agarose gel electrophoresis method. DNA, being a negatively charged molecule, moves towards a positive electrode anode. This process is repeated with vector DNA also. So that is the cutting second step. It is done with the help of the endonucleases or exonucleases depending upon the situation, with the help of the agarose gel electrophoresis technique, and then when it is moving towards the positive electrode pole, mostly that part is over. Then comes the joining of DNA molecules of source and vector involves several processes. Cutout DNA or gene of interest from source DNA and a cut vector with space or mixed with a ligase is added, then resulting in the formation of a recombinant DNA. So see how this is done. This is a molecule A and that is a molecule B. In molecule A, the cut is made here and the cut is made. So this is a palindromic method, I have already told you. So this is the nucleotides are getting repeated from the 5' to 3' and the 3' to 5'. This is a G, this is a G. The next one is a G. Then A adenine, then you have got adenine. T, T, C, C and C, C. So this, when it is moving, it is called, it will be moving in that direction. Okay. Now in this one, it will make a cut here and it will make a cut here. Now this becomes the sticky ends for this. This is called as the hangovers. This is the hangover and this is the hangover for this. Now this will come and then attach itself. This is done with the help of a ligase. Now this has become the recombinant DNA. Now how this amplification is done? Amplification of a gene of interest using the PCR, that is a polymerase chain reaction. PCR stands for polymerase chain reaction. Multiple copies of the gene or a DNA of interest is synthesized in vitro using two sets of primers, small chemically synthesized oligonucleotides that are complementary to the regions of the DNA and the DNA polymerase enzyme. This enzyme extends the primers using the nucleotides provided in the reaction and the genetic DNA as the template. So this is the method of preparing the polymerase chain reaction. So multiple copies of this gene of interest is synthesized in vitro. In vitro, it is normally done in vitro using two sets of primers. The small chemically synthesized oligonucleotides that are complementary to the regions of the DNA and the DNA polymerase enzyme. See, this DNA is the wanted gene. This gene, which is shown in the red color, is the wanted gene that is incorporated here. Okay. So in the first cycle, 2 to the 2 square is the number of the copies made. See, 1 divided into 2, it is called 2 square, 4 copies. Then in the next one, 2 cube, that is 8 copies are made in the second generation. In the third generation, 2 to the power of 4, 16 copies are made. And in the fourth cycle, it is 32 copies. 32 copies, 2 to the power of 4. Two into 2, this is the first generation, second generation, third, fourth, sorry, 2, so becoming 4, 8, 16 and 32. It will be multiplying in that way. Okay. 32 copies are made. And see, in 2 to the power of 36 generations, How many number of the copies it would have made? 68 billion copies. Cannot even imagine. What is the 68 billion? 68 billion? No? 68 followed by 9 zeros. This is the 68 million and this is 68 billion. Okay? So many number of copies, unimaginable number, a very big number. So many copies of DNA is made at the end of 36 generations. So this is what we call as an amplification. It can be multiplied by any number. I mean, you can achieve any number. Process of replication of a DNA is repeated many times. Segment of a DNA can be amplified to approximately 1 billion copies are made in 30 cycles. See? In 30 cycles, you will be able to make 1 billion copies. This amplification is achieved by use of a thermostable DNA polymerase from a bacterium, Dermis aquaticus. So, Dermis aquaticus is the name of the bacterium, and you are able to get that thermostable DNA polymerase. It remains active during the high temperature induced denaturation of a double DNA. So, what is a denaturation? This is a denaturation. When a DNA is in a twinned form, it is twinned in this. When the two DNA threads are separating out, it is called a denaturation. This is a denaturation. It is getting separated. See? So, for that very high temperatures in it, which remains active during the high temperature, induced denaturation of the double stranded DNA, and this is being provided by Dermis aquaticus, which is a bacterium. This amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Then the fourth step. What is the fourth step? This is the insertion of the recombinant DNA into the host cell or organism. Now, ligated DNA in vector is introduced into a recipient cells after making them competent to receive. That's more important. So, the cell should be made competent to receive. If it is not competent to receive, then at this stage, you have to stop the experiment at this stage. The cell should be made competent to receive the DNA. The ligated DNA in vector is introduced into the recipient cell after making them competent. But there are different methods to make them to competent. See, for sometimes you have to dissolve the cell wall, because the cell wall may not be allowing the DNA to enter in. So many problems will be there. So, first it should be made competent to receive. They take up a DNA present in its surroundings. When a recombinant DNA bearing the gene for resistance to an antibiotic is transferred into E. coli cells, these cells become transformed into ampicillin resistant cells. See this point? When a recombinant DNA bearing the gene for the resistance, it is bearing the resistance for what? It is bearing the resistance for ampicillin, which is an antibiotic. It is transferred to the E. coli cells. These E. coli cells will become now resistant to the antibiotic. Okay, and these cells become transformed into an ampicillin resistant cells. If these transformed cells are spread on agar plates, only transformants will grow, because you see it is resistant to the ampicillin, okay, while untransformed ones will die because of the absence of the ampicillin resistant character. This gene is called as a selectable marker. So how you will be selecting the cells? You have to spread them over the agar plates. While you are selecting, when you are spreading them over the agar plates, the transformed E. coli will grow, because they will be containing the antibiotic resistance. That which have not taken the new genes, that means the bacteria which are in the untransformed state, they will all die because of the absence of the ampicillin resistant character. This gene is called as a selectable marker. Then the next step is obtaining the foreign gene product. When a piece of alien or there's a foreign gene DNA in the vector transferred into host cell, the alien DNA gets amplified. Ultimate aim in recombinant DNA technology is to produce a desirable protein. The inserted recombinant foreign DNA is expressed under appropriate conditions. This involves understanding many technical details or technical skills. Okay, now we'll go into the details of this. How are you going to obtain a foreign gene product? When a piece of a foreign gene in the vector transferred into a host cell, the alien DNA gets amplified. Okay, the alien gene that you have transferred is getting amplified. Ultimate aim in recombinant DNA technology is to produce the desirable protein. That is our ultimate aim. Okay, we want to get the desired protein. The inserted recombinant foreign DNA is expressed under appropriate condition. That's very, very important. So you have to provide the conditions where it will be able to express itself, amplify itself. This involves understanding of many technical details. Okay, those people who are really involved in this, they will be, it may be required for them, not for you exactly, because it's all the technical aspects. See, you have got a retrovirus, virus membrane and fuses the host cell membrane. So first it is a comment. This is the way how it is entering. Finally, this is dividing into 2 and then it is made into 4. The viral genes are replicated whenever the cell is dividing. Okay. Now, this is another slide which shows you beautifully about the amplification. Now, this DNA fragments from a donor cell you are getting. These are all just examples of the DNA fragments A, B, C, D, E and then F. All these things, they are all only the just examples. Now, this one, you have got the recipient cell, you have got these DNA fragments. Now, you want to transfer this one, capital N, capital B, and then DNA fragments from a donor cell, recipient cell takes the donor DNA, chromosomal DNA. Now, this comes from here and then it becomes a part of it. Now, what happens? This B gene is incorporated into this original gene. So, this recombinant DNA occurs between the donor DNA and then the recipient DNA. After these genes are induced to express a target protein, the latter should be produced in large scale. That is the main game. So, when they are induced to expression of the gene is very important. If they are able to express successfully, then the next step is to produce those things in a very large scale, in a mass scale, in a commercial scale. The protein encoding gene expressed in a heterologous host is called as a recombinant protein. Okay, just like a recombinant DNA, it is called as a recombinant protein because it is being produced as a result of the recombinant DNA technology. The cells harboring cloned genes of interest may be grown on a small scale in a laboratory, and the culture derived may be used for extracting desired protein and then purifying it by using different separation techniques. Okay, that's mostly we have come to the end of this discussion because this is we are in the last stage. The cells harboring the cloned genes, the cells harboring the cloned, we have taken a particular gene and then we have cloned it. The cells are there carrying this. Now these cells which are carrying our desired genes are cultured and then they are used for extracting the desired protein, because it is with that aim we have transferred the gene. You wanted to get a particular protein to be amplified, so that gene you have removed, then you have incorporated into a DNA, made them into an rDNA, and then you have amplified it, and then it is being now cloned, and then you are getting it. Then after the production of the protein, it could be purified and then using different techniques. This purification involves a lot of techniques. So you have to use a suitable different separation techniques because it will be mixed with all the other proteins. You have to develop your own method for selecting or separating the protein that you look for. In a large scale production, cells can be multiplied in a continuous culture system, whereas used medium is drained out from one side while fresh medium is added from the other side to maintain cells in their physiologically active or exponential phase. This leads to higher yields of a desired protein. Naturally, you see, you have got a experimental vessel. Now in this you want to have a continuous production. So what will you do? When it is in a large scale production, the cells can be multiplied in a continuous culture. The used medium, you are using a medium, and this used medium should be drained out because it is used already. It is the medium has been used out. So the used medium is drained out from one side and a fresh medium is added, so that it is the reaction is continuously going, is taking place in a regular form, so as to maintain the cells in their physiologically active condition. See, if you are not removing the medium, then the medium will lose its potency, will lose its physiology. So you have to remove the medium, which has become useless, and then you have to add the fresh one. So this leads to the higher yield of the desired protein. So this step is also very, very important. So now you have got to, for this purpose you have got what are known as the bioreactors. If you are going to produce a protein in a very large scale, then you must employ what are called as the bioreactors. These are the vessels in which raw materials are biologically converted into specific products to be enzymes using microbial, plant, animal and human cells. So you can multiply microbial organisms, plant cell, animal cell, or a human cell, or a bacterial cell, anything for that matter. Okay, if you want to produce a large then you must take the help of a bioreactor. What is this bioreactor? It contains a large volumes. It's 100 to 1000 liters of a culture can be processed in a bioreactor. So different bioreactors are there, small size, medium size, large size, like that. Different reactors are there, which can, the wide range is also there to hold 100 liters, 1000 liters, etc. So 100 to 1000 liters of a culture can be processed in a particular time. It provides optimal conditions for getting desired product by providing optimum growth conditions, temperature, pH, substrate, salts, vitamins and oxygen. So everything should be maintained properly. This is a very simple terms we have told. Optimal growth is achieved by providing all the necessary conditions. The temperature should be maintained, pH should be maintained, then substrate should be made available, then salt, vitamins, everything should be maintained in a bioreactor perfectly. Then only there will be a continuous, the product will be there. You can get the best output. See, the last step in this is what is called as a downstream processing. After biosynthesis of the product, it has to be subjected through a series of a process before it is ready for marketing. The process includes separation and purification. This is very, very important. So you have to separate the protein, should not get contaminated. You have to separate the protein that you want to be marketed. And then of course the purification is also very, very essential. Then you have to separate and then purify. Collectively called as a downstream process. So the separation and purification is collectively called as a downstream processing, which is the last stage in the marketing. Product has to be formulated with a suitable preservative. That's also very, very important. Such formulation has to undergo through the clinical trials as in drugs. A new protein you have manufactured by using a bioreactor. Now you have separated, you have purified it, everything is over, and then you have also added the preservative. Now that's not the end of it. Now it has to undergo clinical trials. You have to give it to the animals first, and then you have to find out what effect it is having on the animals. Then it has to be, everything has to be proved by statistics that it is having the best effect, the effect that you wanted to have, and it is not having any adverse effect. Perhaps you have to choose a certain first animals, about 100 or 200 or 300. You have to give the desired protein or the desired drug or whatever you have done, and then that drug is giving out its best performance, and it is able to control the disease. Everything you have to prove, and it is not having any side effect. All these things have to be proved statistically before it is coming to the market. These are what is known as the clinical trials. Then after giving it to the animals, you have to try it with the human being. Of course, getting the permission from the individuals, from the government that you are doing. Then you have to give it for the, you have to get the consent letter from the patients, and then you have to administer this drug, and then you have to find out how much of effect this drug is having, whether it is having the desire, is able to give you a desired result, or whether the drug that you have prepared has become totally useless, and whether it is going to have any side effects, and whether it's not at all having any side effect. Everything has to be clinically proved, and then only it could be released into the commercial purpose or marketed. Then strict quality control testing is also required. Otherwise, the whole human population will become the whole thing becomes a question mark. Nowadays, what happens? The problem comes only in this. No strict quality control is there, because everything has become business. Everything has become business. They take a very small group of people, they administer it, and it is also not the quality of the medicine is not, the efficacy of the medicine is not proved beyond any doubt. And then very strict measures are also not applied. Then just like that, it is coming to the market. Then after 6 or 7 years, we find that a particular drug is having some side effect, and it is having some adverse effect also sometimes. These problems also we normally encounter, because the strict quality control is not there. Okay, so downstream processing and quality control testing vary from product to product naturally, because if it's going to be a drug, if it is going to be a medicine, then it has to be administered on the patients. Then a number of clinical trials has to be given for that. If it is going to be some other thing, then it all depends upon what product you are trying to bring to the market. Okay. Then a little bit of information I am going to give in this lecture about the biotechnology and its application. Now we have, I was just telling you that you have got a desired gene with you that you have got from something else. And now you are combining that gene into an organism, into a plant, into an animal, or into a bacterium, and then you have amplified that gene, and then you have modified an organism, what's called a GMO, genetically modified organism. Genetically modified crops, genetically modified plants are also there. So you are manipulating the genes, and then you are bringing out a plant or an organism for that matter, and then these new organisms that are coming out. Now this is what is called, this is what we wanted. We wanted to make the plants and animals to give the best result by the biotechnological processes. So this application part of the biotechnology, a little bit I am going to teach you. It deals with the large scale production of biopharmaceuticals using genetically modified organisms like microbes, fungi, plants and animals. Biotechnology application includes, these are all the areas in which the biotechnology is working now: therapeutics, diagnosis, genetically modified crops for agriculture, processed food, bioremediation, waste management, energy production. So these are all the areas where biotechnology is spreading its wing. Therapeutics. So therapeutics, it includes so many pharmaceuticals. It's a very broad area, therapeutics. So many medicines has now come to the market because of in the case of diabetes, and for different type of cancer, and for some dreadful diseases. Now genetically modified medicines are coming, drugs are coming to the market. So this area is the therapeutics. Diagnosis. For diagnosis, see, for example, if a woman is carrying a defective baby, genetically defective baby, 21st Down syndrome, or so many syndromes will be there, and this could be diagnosed beautifully today with the help of a biotechnology. Even at the 4th month or the 5th month, we are able to carry out this diagnosis. These are all very much beneficial to the human being, and if we find that there are some problems, it could be dealt with properly, with the proper care. So in the diagnosis also, biotechnology is playing a very vital role. Genetically modified crops we are able to produce in agriculture. Hundreds of crops we have made, which are genetically modified. Then in the food processing, new preservatives are coming to the market with the help of biotechnology. So preservative processing, in the food processing alone, biotechnology is doing a tremendous work. Then bioremediation is also mostly what is known as therapeutics. Bioremediation, and then waste management. You see, it's a very important thing. In the waste management, which is playing a very vital role, different type of microbes, bacteria, viruses, and bacteria, fungi, and so many microbes are there. These microbes are able to bring about a lot of change in the waste management. They are able to break down some of the waste materials and then convert it into the usable form. The efficiency of these microorganisms in waste management is beautifully amplified with the help of this biotechnology. So biotechnology is playing a very important role in the waste management and then in the energy production also. So these are all the areas where biotechnology has proved to be very beneficial to the human beings or mankind. rDNA has been becoming more important in this century as genetic diseases have become more prevalent and agricultural areas are reduced. Some areas where rDNA will have an impact. See, rDNA has been becoming more important in this century as genetic diseases are becoming more prevalent. Genetic diseases are becoming more prevalent. So, genetic diseases could be identified at a very early stage in the diagnosis, which I was remarking. Then in the agricultural areas also, it is playing a very important role. Agricultural areas are getting reduced. So when the agricultural areas are getting reduced, you can improve the quality of the crop. You can the output of the crop could be made more by using these technologies. See, better crops. In what sense you call them as better crops? They are drought and heat resistant crops. Recombinant vaccines we are able to produce. Hepatitis for hepatitis B, you are able to have a recombinant vaccines, not the normal vaccines or vaccines. Recombinant vaccines, just like a rDNA or protein, you got or vaccine. Recombinant vaccines. Plants that produce their own insecticides. You see, now this a particular plant is there, A, and this plant A is being attacked by an insect called small a. Let's imagine this small a insect is attacking this. Now what happens is this plant A is given a gene by recombinant DNA technology so that it will be able to resist this insect. Okay, so genetic resistance is infused. Genetic resistance is given in the form of the gene to the plant A to repel the insect A. See, that is the one. Plants that produce their own insecticides, you need not spray a chemical insecticide over it. The plant itself will be secreting its own insecticide inside its system. How it is able to do? It is because of the gene that it has derived. From where that plant has received that gene? It is by the DNA technology. Okay. So, prevention of a cure of the sickle cell anemia. Typical examples I am giving to you. Sickle cell anemia is a genetic disorder. A man having, a baby having this genetic combination will not be normal, and this one will also be normal, and if both the alleles are recessive, then this baby is turn out to be a sickle cell anemic case that you would have studied. The RBCs are becoming sickle in shape. That is why it is called sickle cell anemia. You have got a different varieties of a sickle cell anemia. Sometimes the sickle is like this, sometimes it is too much, sometimes in a very acute case the sickle becomes like this. RBC is becoming sickle in different grades. Okay, for this sickle cell anemia we are able to, a cure has come because of the DNA technology. Prevention of clotting factors, another very beautiful thing. See, clotting factors. There are problems called what is known as hemophilia. You would have heard. See, for a normal man, in a typical wound, when it is bleeding, for the coagulation and for clotting, how much time is required? It makes normally 2 to 3 minutes. Normally, if the wound is not involving a vein cut. If it is not involving a vein cut, normally it will get coagulated within 2 to 3 minutes, more than sufficient. But for some people it takes hours together. So they have got what is known as they are called hemophilias. The coagulating system is failing in them. The blood is not coagulating. So that is now that clotting factor, that disease is being taken care of today by means of DNA technology. And then prevention of cystic fibrosis, dangerous, a dreadful disease for the women. Fibrosis, uterus fibrosis is the maximum problem that is. See, in today's world, with the changing scenario and in the changing lifestyle of the women, we find that at least 60% of the women are having the fibrosis. And there is when a girl or a woman goes to a doctor complaining about uterus pain, he says uterus fibrosis is there, and today everybody is having that problem. Don't worry, you can take some medicines. Just like we don't bother about the diabetes, because it has come to mostly all the people, so we don't bother about it. Similarly, fibrosis, uterus fibrosis, it has come to most of the women. So it has become a very common, just like a common cold, it has become a common disease. So prevention of that is very important, that we are fighting. Then production of insulin, this is another very important thing. For the insulin production, we were depending only on what is called as pancreas of the pigs which were slaughtered. But now we have got the E. coli bacteria which are secreting the insulin for you. Then production of recombinant pharmaceuticals. Then germline and somatic gene therapy. So these are all some of the areas where we can take the help of the biotechnology for improving the quality of the life of the man. Application agriculture, out of the three options for increasing the food production. So agrochemical based agriculture, this is one type of agriculture. Agrochemical based agriculture, where you add only mostly all the insecticides, pesticides, manures in the form of NPK. So your agriculture has become mostly chemicals. Then organic agriculture is there, when you add organic manures. Then genetically engineered agriculture is there, genetically engineered crop based engineered. So the last option is better. Genetic modification of a crop has made crops more tolerant to abiotic stresses, cold, drought, then salt, heat, etc. Reduce reliance on chemical pesticides. Now you don't rely on the chemical pesticides. Reduce, not totally. You can't totally avoid it. You have reduced the reliance of chemical pesticides. Then helped to reduce the post-harvest losses. So that's very important. Increase the efficiency of a mineral usage by plants. Then you can make a plant to take more minerals. Increase the efficiency. You can improve the efficiency of a plant by taking more minerals. Then enhance the nutritional value, food value. You can increase. You can increase the protein value. You can increase the vitamin value. See, all these things you can increase by means of what is called as a biotechnology. Medicines. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 are marketed. These don't induce unwanted immunological responses, as in the case of products isolated from the non-human sources. They can be produced in mass scale. So the benefit is they don't have unwanted immunological responses, which is a problem in the traditional system. But today that problem is solved. See how the bacterium is modified to produce its own. There is a bacterium and you have got a plasmid cut enzyme. You have incorporated a gene there. This incorporated gene is made to amplify. The bacterium divides and then it is producing the insulin. The E. coli bacterium is producing the insulin. This insulin you have inserted from a human gene for insulin production, and this is being amplified, and then you are getting the insulin. Gene therapy. This is a collection of methods that allows a correction of a gene defect that has been diagnosed in an embryo or a child. Correction of a gene defect involves the delivery of a normal gene into the individual or embryo to take over the function of the gene and compensate for the non-functional gene. Beautiful. See, this defective gene is removed. Normal. What is a correction of a gene which is a defective by a normal gene, and then it becomes you can remove the defect from the fetus. Here genes are introduced into a person's cell and the tissues or then it is treated for the diseases. Gene therapy for the ADA asset. So this is the T cells present in the man, and then these T cells are taken, and then human ADA DNA, and then the infection is there. Now these T cells are taken. T cells carries the human ADA CNA. Now this is a corrected ADA gene. So this is the corrected ADA gene, and then this corrected ADA gene is made to amplify, and then these amplified genes are infused back into the patient. So now this man is not suffering from the ADA. Okay. So the genes are taken, it is corrected, and then it is introduced into the man once again, so that he is not suffering from the disease anymore. Gene of interest. Can take any gene of interest. Gene would could remain unwanted to undergo the mutagenesis. Then this is introduced into the vector. Then the plant, bacteria, yeast, and then mammal, anything it could be introduced, and then it could be used for protein purification. Further modification is there, then formulation is there, final pharmaceutical products. These are all some of the areas in which biotechnology can really help you. Biological products are as follows: human protein alpha-1 antitrypsin for treating the emphysema. Then PKU, there is a biological product for treating the phenylketonuria. Human protein enriched milk 2.5 grams per liter from the first transgenic cow, Rosie, in 1997. So it is a 2.4 grams per liter from a transgenic animal. It contained alpha lactalbumin and was a more balanced product for the human babies than the cow's milk, and the normal cow's milk. Oil producing soybean was transformed into transgenic plant capable of producing a wide range of industrial lubricants, cosmetic compounds, and detergents that are biodegradable. So another beautiful area. See, the different type of industrial lubricants, then cosmetics, compounds, and then these are all detergents. These are all very harmful to the human being. So, and by introducing this biotechnological process, we have made these products as biodegradable. So in the pollution you will be reading that that a particular structure is a biodegradable and non-degradable. If a compound is going to be a biodegradable, then it's not adding anything more to the pollution. So some of the compounds are made biodegradable very easily. Plants are made to synthesize many novel substances, including functional human antibody fragments. Gene forming is another very important area. Use of transgenic plants as bioreactors or factories for production of specialized chemicals and pharmaceuticals. Then production of human proteins in the form of hormones. In the seeds of transgenic seeds, production of polyhydroxybutyrate, a biodegradable plastic in tissue granules in a transplanted weed. There is a mouse-eared crust. So these are all some of the areas where you can where you are really introducing a compound into the system or into the nature, replacing the already existing one which are detrimental to the human. So perhaps in my last slide, these are all some of the transgenic plants that we are having today. I am dividing into dicots and monocots. In dicot, some of the transgenic plants are Nicotiana tabacum, Beta vulgaris, Glycine max, Helianthus annus, Solanum tuberosum, Gossypium hirsutum, Solanum melangena. So, Nicotiana tabacum, you know very well, tobacco. Beta vulgaris, a beetroot. Glycine max, it's a type of beans. Helianthus annus, a sunflower. Solanum tuberosum, potato. Gossypium hirsutum, a type of cotton. And then Solanum melangena, there is eggplant. So these are all some of the plants in dicots where we have introduced the plant genes and then we have made them into the transgenic plants. Into the monocots, we have got the asparagus species, Oryza sativa, there is a paddy, the maize. That is what is known as the maize. In English, it is written as maize, M-A-I-Z-E, not writing properly. The maize. Then Avena sativa, which is called the oats, Triticum sativum, wheat. So these are all some of the monocotyledonous plants where the transgenic plants have been introduced into the nature. So this gives you a beautiful picture of how biotechnology could be made more useful to the humankind. Okay. So what is biotechnology and what are the different tools involved, what are the processes involved, how a biotechnology experiment can be carried out, how the biotechnology process could be done very successfully, and how it could be made more useful to the humankind. These are all certain areas which we have discussed very beautifully in some of our lectures. Okay, fine. After hearing this, you may be able to understand how much beneficial biotechnology is for us. Okay. And then one more lecture is there wherein I will be discussing about the application part of the biotechnology in a separate lecture. Okay, thank you. Good. Now you are listening to my lecture about the biotechnology and the related areas. If you could pause on your comments, if you could pause on your information, see, if you want to add something more, you can do so beautifully. Be nice. Thank you.